All right. Hi, everyone, and thank you for being here today. I'm Brianna Haran, Vice President of Clinical Sales at Galaxy Diagnostics. Many of you are seeing these kinds of cases that we're going to discuss today every week. Patients with persistent symptoms, complex histories, and results that don't quite give you a clear answer. And at some point, the question becomes, what am I missing? That's really what today is all about. At Galaxy, we spend a lot of time thinking about those gaps where clinician suspicion is high, but the data doesn't fully connect. And that's especially true when it comes to vector-borne disease. So I'm really excited to bring this conversation to you. It's a pleasure to introduce Dr. Jennifer Miller. Dr. Miller is our Vice President of Clinical and Scientific Operations and our CLIA lab technical supervisor. She has over 20 years experience in vector-borne and zoonotic pathogens, including Babesia, Bartonella, and Borrelia. She's been deeply involved in developing and advancing the diagnostic gaps that help uncover these infections, especially in cases where they're easy to miss. What I appreciate most about her perspective is how clearly she connects the science to what you're actually seeing in your practice. Jen, I'll turn it over to you.
Well, thank you. Thank you so much for the kind introduction, Brianna. Grateful to be able to have the ability to speak with you all today. Let me share my screen. Okay. All right. Hopefully everybody can see. So today we're going to talk about Babesia, the black box of vector-borne pathogens, with an emphasis on uncovering what we're missing, but also, most importantly, why it matters. These are just slides about us. So why does this matter clinically? You're already seeing patients that are presenting with chronic fatigue, neurological symptoms, dyspnea or air hunger, multi-system involvement. And this clinical experience often includes multiple prior evaluations, results that may be negative or inconclusive or difficult to interpret, and of course, a persistent symptom burden. And despite testing, symptoms may remain unexplained and the clinical clarity remains limited. So, case study one. This is an example of when the clinical picture does not resolve. So, this is a patient that was a 35-year-old male from Connecticut, long history of complex illness, including prior diagnoses of Lyme and Bartonella. The clinical presentation consisted of cognitive dysfunction, intermittent rage episodes, and multiple psychiatric admissions. Patient had extensive serology and fish testing, but I will note no previous Babesia diagnosis. So samples were submitted to Galaxy for testing. A 3-day draw digital PCR was selected, and we were able to detect Babesia. I will note that the serology was negative on this same blood draw. So this sort of illustrates a bit of clinical testing, clinical tension where history suggests infection, but testing did not consistently confirm it. So this sets up a conundrum. If Babesia is present, why don't we consistently recognize it or detect it? And so this is sort of where the title of the talk comes from. You know, what do we mean by the black box? And so what we mean is that the black box exists where clinical suspicion is present, testing has been performed, but there are unexplained results that do not fully explain the patient or what they are experiencing. And this happens because there's four distinct layers that may not align. There's the clinical presentation, the pathogen biology, the available research, and the diagnostic detection. So for symptom presentation, kind of breaking these down one by one, as we said at the very beginning, these patients are presenting with some common clinical features. They have fatigue, it may be persisting or relapsing. It has dyspnea. I'm struggling over that. Air hunger, night sweats, cognitive dysfunction. And this leads to clinical variability where symptom intensity fluctuates, presentation differs across patients. But the key here is that the observed patterns may not match the provider's expectation or experienced clinical judgment and decision making. So symptoms can overlap. You know, what do we mean by this? These types of symptoms commonly appear across not only Babesia, but also Borrelia, Bartonella, and other chronic inflammatory conditions. And because the symptoms are not pathogen specific, it makes differentiation based on symptomatology alone limited or in some cases very difficult. So it sets up a clinical challenge. Babesia may be misattributed as Bartonella, Borrelia, or any one of a number of non-infectious chronic conditions due to the contributing factors of overlapping symptom patterns, lack of distinguishing markers, variable provider experience. And this leads to inconsistent clinical suspicion and variable diagnostic pathways. And so what happens is that patients get stuck. And so where they get stuck is that their symptoms persist despite evaluation, and their diagnostic experience is a potpourri that may include positive serology that's difficult to interpret, negative or inconclusive molecular results, and discordant findings that exist across various methods. And so this leads to uncertainty as to what the next steps should be for these patients and also reduced diagnostic confidence. So, in our second case study, this is an example of when detection does change the picture. So this patient was a 44-year-old male residing in Oregon, presented with progressive neurological and multi-system symptoms. The patient had had extensive prior testing and had sort of a treatment refractory clinical presentation. And so Galaxy was able to detect both Babesia and Bartonella with our molecular testing. And so the insight here was that this patient had a co-infection that was missed in prior testing and prior to multiplex testing. And so the implication of this is that what you detect depends on the methods that you select to look for the pathogen. So there's also a bit of a biology problem that can sort of be lurking underneath with all of this. And, Babesia, as many of you know, is an intererythrocytic parasite and infects red blood cells, exists within circulating erythrocytes, but detection of it often depends upon parasitemia. And this sets up another challenge because parasitemia may be low or intermittent, which what that means is that the organism itself may not be present in every sample. So this sets up a bit of sampling bias in that detectability can vary across time. In other words, when do you assess the patient and also the draws? When are you drawing the blood and how many times? There are also some limitations with Babesia. Babesia is well studied in animal models, but our human specific understanding remains more limited. And that's because the amount of funded research, human research in the United States is very, very limited. I would love for NIH to fund more basic research into Babesia. And even worldwide, a lot of people that are studying Babesia are sort of studying it in animal models or as a problem that is agricultural in basis, and that's very real, but we do need more human specific information. Its pathogenic behavior is not fully defined. Clinical frameworks remain incomplete. And so interpretation relies on evidence that is continually evolving and changing, but yet is incomplete. So in the diagnostic context, Babesia testing may include blood smears. This is the most tried and true, and it primarily works for acute babesiosis, where you're going to have a lot of parasites in a blood smear, and this is mostly done in a hospital setting. FISH, or fluorescence in situ hybridization, is an emerging technique that providers may elect for FISH. Serology, this is looking at immune response detection, antibodies that are raised against the parasite. And then PCR-based molecular methods, which are a way of sort of directly detecting pathogen DNA. Each of these methods have pros and cons, and because of that, they're not interchangeable. And so detection is not one moment in time. It's influenced, and I alluded to this, by the timing of sample collection, what the parasitemia level is at the time of the blood draw, how many samples you're able to collect from the patient, and the sensitivity of the method. And so why do I say this? Single time point testing may miss low level infections, and detection may vary across time and sampling. So what's the implication of this? A negative result does not always mean that the organism is not there. It may reflect limitations of sampling and detection. So I want to talk a second about interpretation of serology. As I said before, serology detects the host immune response. And there's a lot of benefits to this, but I think you need to understand the limitations. Because you're detecting a host immune response, it may indicate that the person's been exposed, but it may not necessarily indicate that the person is currently infected with the pathogen. And so serology requires interpretation within the clinical context. These diagnoses are clinical diagnoses, and the provider has their vast clinical experience and clinical judgment and history with the patient. And all of that goes hand in hand with testing. But I do think it's instructive to really think about, what does a positive result actually represent? What is it telling us? And so with direct detection, molecular methods are detecting pathogen nucleic acids, so DNA or RNA, and it's indicative of organism presence or presence of a pathogen in a sample, because if the nucleic acid is there, it's a signature. It's sort of like the pathogen was here. It can be indicative of parasitemia at draw, because there are molecular methods that measure and quantify, the burden of the parasite genome and copy number. And the assay design matters. How the assay is designed and the methodology really matters. And so a limitation, as most of you know, with molecular testing is that absence in a sample does not mean absence in a patient for some of the reasons that we just alluded to. So I want to talk about assay variability. So not all assays are equally designed. Sensitivity varies by methodology. How is the assay being performed? Is it quantitative? Is it qualitative? How is it validated? What types of quality controls are you running with the assay? How much clinical data do you have with the assay? Platform differences, detection capability differ across different platforms. You have qPCR versus digital PCR. What those can do from a platform really varies. Some assays are better suited for low abundance organisms. And so because of this, if you get differences in results, it may be due to differences in how the assay was designed. And so kind of going into this a little bit deeper, why do results differ? Well, different methods are measuring different biological signals. So as we alluded to, parasitemia is a measure of the organism in circulation. You're measuring genome copies or parasite copies. Pathogen presence, you're looking for a signature. You're looking for a DNA or RNA that's derived from the pathogen, so that's a signature. Post-immune response, you're looking at antibodies to the pathogen that are being raised by the human. So differences in results absolutely reflect differences in what we are measuring. So again, what does that result mean? Um, and so for as we think about this at kind of the population level and what we see clinically, patients are presenting with chronic ongoing complaints that are resisting sort of the standard diagnostic treatment protocols and explanations. You get laboratory and clinical findings that are frequently contradicting each other. So it's very hard to give the patient a definitive diagnosis. The clinician's frustrated, the patient is frustrated. It's kind of like you're trying to solve a puzzle when a piece is missing all the time. This leads to ongoing diagnostic uncertainty because despite thorough workups, lots of testing, many cases remain unresolved. And so patients don't have answers, and it's hard for clinicians to have a clear path forward. But there's hope. In emerging research, Babesia DNA was detected in 24% of patients with chronic fatigue and neurological symptoms using serial molecular approaches. And so this was striking because it allows a bridging of individual clinical observations to population level dynamics, and more work is needed to dig into this deeper and really kind of uncover what the prevalence is and what our ability is to detect these pathogens. So if you look at the population level statistics, about 3 million adults, or about 1.3% population in the US, have reported a Babesia diagnosis at some point in time. On an annual level, the CDC is reporting 1,800 cases annually. But there's evidence that suggests that this annual number is very underestimated and underreported. And we get sort of glimpses of this by looking at insurance data. And when we look at the insurance data, it suggests that the number is closer to 25,000 per year, which means that the incidence is a lot higher than what the CDC is able to capture with their reporting mechanisms. And we are learning through our case studies and communicating with providers and learning about their experience treating these patients that chronic fatigue is present in a significant portion of Babesia cases. And this has also been reported by others in the literature. And a subset of these patients experience prolonged illness. In other words, moving past sort of the paradigm that Babesia is only an acute manifestation. We're seeing that that's just not always the case. So this sets up kind of a complexity that's arising from symptom overlap with other pathogens, a parasitemia burden that may be low, limited human research, variability between assay validations, assay platforms, and methodology. And then all of this, leads to the final issue, which is challenges in interpretation. So moving on to our third case study, this is an example of sort of broadening our, clinical patterns and sort of our what is a typical patient, although I would argue with these particular infections, sadly, there is no typical patient. This patient was a 34-year-old female from North Carolina who had no clear evidence of tick exposure. Patient presented with hormonal irregularities, fatigue, dizziness, migraines, but no sort of classic Babesia symptoms. And so Galaxy detected Babesia via our 3-day draw digital PCR. And I will note that the IFA serology in this patient was reactive for Bartonella. And so in this case, detection revealed Babesia in a non-classic clinical presentation. So the difference between clinical experience, reported cases, and research findings may reflect differences in detection methods, differences in clinical recognition, and limitations in reporting systems, all kind of setting up this idea that what we detect is influenced by what we are able to measure. And so, kind of back to this kind of a common theme, what does the method actually allow me to see? What do the results mean? And this impacts clinical decision making because it comes down to what is the test measuring? What can it detect? And then what uncertainty remains after you have that test result? What is it not answering? And so, again, the challenge isn't just in detecting Babesia, but how do you interpret the results when you have symptom overlap, evolving research, sort of the biology of the organism makes it hard to see because it may present at very low or circulating parasitemias, and methods for detection, all of which have their utility, are not equivalent and interchangeable. So kind of taking this back to the patient, you have a situation in each of these three case studies where we had persistent symptoms. Standard approaches did not provide a clarity for either the provider or the patient. And what was able to change is when ultimately it was sort of uncovered that these patients had Babesia, understanding the biology of parasitemia, interpreting discordant results, and selecting a variety of detection methods definitely helped. So the outcome is not just dependent on whether Babesia is present, but what do you do with that information? How is the outcome evaluated and interpreted is absolutely critical. Thank you.
Thank you, Jen. That was great. Would like to open it up for any Q&A that you guys may have. Feel free to, at the bottom of your screen under More, you should be able to select the Q&A. We did have somebody ask about receiving the recording and the slides, and yes, everyone will be receiving both. Takes us a few days just to edit the pauses and things from the video, so if you can all be patient and give us a few days, that would be great, but you will all get both recording and slides. All right, we'll leave it open for another minute for questions. Here we go. All right, Jen, do you see spleen issues with babesiosis? Although she typed in, thank you, Katka. I'm not going to say this correctly. Splenomegaly?
Phonomegaly. I mean, you certainly can. As to whether the samples were being submitted come from patients that exhibit that, that's information that we would only be able to have if the provider discloses it. I will say for a lot of our case studies, not necessarily, just because a lot of the cases that we're seeing are from patients that have kind of chronic fatigue or almost skin tingling as a major symptom. That's not to say that individuals with Babesia can't have splenomegaly. We certainly know that's a major manifestation, but we're definitely seeing some cases that are more atypical in the sense that chronic fatigue and tingling or neuropathy are common things that we're seeing.
This is a question I get asked often, Jen. We have a question of, do you have any estimate of the sensitivity of the current Babesia or part of the BBB direct detect digital PCR for chronic babesiosis on one blood draw, or in general for chronic babesiosis? And then also, what about for one blood draw?
So the way I can answer that is to say that clinical utility of our assay is ongoing. We're continuing to gather clinical data to try and compile by disease state what the sensitivity is. But I will tell you that we have detected very low level Babesia, the level of couple copies per microliter in patients that we were told were chronic. Okay. And I will tell you that, what was the second part of the question again, Bree? Sorry.
So I also wanted to ask if you could just maybe for quickly just cover the difference between an analytical versus clinical sensitivity and specificity, because sometimes I find
Sure.
that practitioners don't know the difference, but
So
really is the difference between the single blood draw versus the triple blood draw.
thank you. That was the piece, but I'll talk about analytical. So analytical sensitivity and specificity is kind of how well does your test perform in laboratory simulated settings. So in other words, you take those quality controls that I mentioned earlier, are very, very important to run with every single assay. And they're usually, blood samples that are spiked with organism material at a known concentration. And you run it over and over and over again to demonstrate, what is the quantitative range that you can detect, with that you're supposed to with positivity, make sure that you're not getting, false positive reactions and samples that are supposed to be negative. Whereas clinical validation is you've acquired well-characterized, known Babesia blood samples that other people have run and vetted as positives. Sort of the equivalent to this in the Lyme community would be the CDC serum panel, or Liz Horn's done a phenomenal job of compiling samples through the Berry Lyme Initiative. Unfortunately, these samples don't exist for Babesia, but Galaxy is working to change that because we believe it's very important to have reference samples. So that means that clinical validation really relies on testing samples from patients in the field in the real world, right? And we're doing that. And that's part of why we engage in this case studies because we receive samples, we test them, but to us, they're not well defined, right? We only know about them if we engage with the provider. And we're lucky that we've had so many providers who are willing to share that clinical experience with us and teach us about these patients. And so we're developing that clinical data and it's ongoing. So I can tell you that we are able to detect very low levels of Babesia on the order of a couple copies per microliter in these patients. I can tell you that we do detect Babesia in single draws, but I'm also going to tell you that statistically so far, and the data is still evolving, we are detecting Babesia at a higher sort of prevalence percentage in patients where we have obtained serial blood draws. Like we've had situations where, and we talked about it in this case series, where we had a 3-day draw and we only detect it in one draw out of those three. Or in very rare cases, I can think of maybe two, where we've detected it in 2 out of 3 of those draws. So again, just based on our experience from what we're testing and what we're seeing, and the serial draw matters. And I will also say that colleagues have published that can make a difference too. Doctors Breischwart and Maggi have published at NC State, in a lot of their work where they're testing large numbers of people that are unfortunately are at suspicion for Babesia infection. They're seeing something similar, that the serial draw matters, and it's our hypothesis that this really relates to the cyclical parasitemia because the Babesia are there, but unlike what you see with an acute microtia infection, it's not this textbook. Everybody has very, very high parasitemia. It is cyclical, and some of this could be due to the parasite lysing and moving to other areas. It could be due to formation of fibrin nests within the microenvironment of an erythrocyte or a capillary, which is going to make it a little harder to detect these organisms because they're wrapped up in host material.
Thank you. What Babesia species are you finding by percentages? For example, Oticoliae, Microti, MO1.
So for us, it's hard to answer that because our testing is at the genus level. Okay. So the only way we would know that is if we were able to sequence, which digital PCR doesn't allow for, or if, providers have other results from other testing. Like I said, there's lots of different testing. It all has its merits. I will tell you from published work, again, from our colleagues, Breischwart and Maggi, that they are seeing higher proportions of Oticoliae and divergens of divergent-like, so say the MO1, in patients that they've looked at versus your standard Microti. And this is an evolving and ongoing area of research. We understand and appreciate that the numbers of these cases are on the rise. There have also been recent publications in the literature of divergens in MO1 being detected in studies that were done out of Yale and elsewhere. So there's definitely this recognition that we are moving beyond Microti into a greater prevalence for Oticoliae and divergens. As I said, I think there's more out there than we even realize.
Thank you. Would you consider testing every CFS patient for Babesia? Which test do you use for the best screening tool if you could only do one?
Oh, that's a very good question. So in my opinion, and again, I'm not an MD, not a clinician, but as a researcher, if you as a provider have a patient with CFS and you feel like with a lot of CFS cases that there's an infection, then I think it's worth your while to test for Babesia. Now, I would say that for those, I understand that cost can be an issue. So I would say that if you have to screen, this is where antibody testing could be helpful. It could be helpful to screen with some kind of serology test or something else to get an idea of has the patient even seen Babesia, right? And then if they have, because you don't know whether it's current or not, then I think it's perfectly acceptable to fold in the molecular techniques.
And just to kind of close the loop on that, what we're seeing from the single draws to the triple draws, we recognize that cost can be prohibitive for some patients. We do get single draw positives, but patients from the data that we've seen so far are 2.5 times more likely to be positive on a triple than a single. And again, triple draw was born because of Bartonella's cyclical nature, but then when we added Babesia to that BBB test, those triple draw Babesia positives are not positive on all 3 days. So we're really dealing with these low level infections, especially once that patient is chronic.
Agreed.
Do you see bilateral or unilateral neuropathy? Sorry, do you see bilateral or unilateral neuropathy with babesiosis?
So we definitely have, again, through our case studies, reports from providers that some patients do have this, but definitely not all.
We do get a lot, specifically the word, because since I'm on these positive cases as well, we hear a lot of tingling. And typically it's not bilateral, it is typically unilateral down a full extremity, like in an arm or a full leg. And we've also had specifically the back of the head. Again, is not a huge study or anything like that, but just sharing with you what we've learned from the practitioners.
But we're definitely open to continuing to, expand our sample size, right? The more we can learn from providers and clinicians that are on the front lines, trying to take care of these patients, that expands our knowledge of the clinical utility of our test.
Absolutely. All right, we've got one last question. Why is it so hard to get Babesia on serology? Is it because it lives in the RBCs? I'm confused
Yes.
why it's so hard to detect it if its
So
home is in the RBC.
that's a big part of it. It also sort of comes down to the type of serology test. And this ties back into we talked about with the limited biology. So if you're going to do an IFA, for example, you've got to be able, whether it be us or anybody else, right? You've got to be able to cultivate that Babesia to make those slides. And since it grows inside red blood cells, it creates a scientific challenge because you can't just go out and pick your favorite cell line off the shelf and grow a flask of Babesia and infect those cells, right? That's so there's an actual scientific challenge because you have to these things only propagate inside red blood cells. So that makes it a challenge, right? To actually get those slides and make them. And you say, well, Jen, certainly in the Lyme literature and even getting closer now with Bartonella, we know there's certain key virulence factors, recombinant proteins, things like that. Can't you just make an IFA based on that? Maybe you could, but again, this is where the literature with Babesia is limiting. There's so little known about the virulence factors of Babesia that a lot of what's in the review articles is hypotheses that are made based upon comparisons to malaria. It's really like the old school SATs.
Mm.
Malaria has all these things, and Babesia and malaria do a lot of the same things. Therefore, we're assuming that these types of things also exist for Babesia. But it's not really known because a lot of the studies have been done in animal models. And then it's not known how do expression of those determinants, where we do know what they are, how does it vary? Is the expression the same in microti as it is in oticoliae, as it is in divergens, as it is in Duncani? We just don't know. So a lot of this leads to really technical challenges in developing serological assays for Babesia. And because we don't know how the expression varies or even overlaps between these strains, you have the question of cross reactivity, right? Because even if you go to a recombinant protein, if you even knew what that was, does Duncani have the same one as divergens, the same one as oticoliae? If yes, you're going to have cross reactivity, right? And so it just creates this is one of my favorite topics, as you can tell. It just creates a big technical challenge that we and others would really love to overcome.
Jen, you mentioned, and I'm just going to tease this out a little bit more because, again, what comes up in a lot of provider meetings, what's a recombinant antigen? Why does that contribute to cross reactivity? Is
Yeah,
Galaxy the same or
recombinant
different?
antigen is just, you identify, and there's lots of different antigens, but I'll pick the easy one. Let's say protein. You pick a protein that and again, I'm going to go back to something where we have more knowledge to draw a parallel, and that's Lyme disease. For example, if you know that you have a virulence determinant that is expressed, I'm going to pick one a lot of you are familiar with. Let's say VLSC or C6. Okay. You can engineer unrelated systems, say E. coli, insects, whatever, to artificially pump out large quantities of this protein, right? And you can theoretically create an IFA with it or an ELISA, okay? And it might be clean, right? You're going to have great specificity. But in terms of sensitivity, if you don't know how well that protein you've made structurally aligns across all the various different species and strains and across all the various places where the protein can show up, then you could have a sensitivity problem and you could have a cross reactivity problem. And so it's very complicated. And so if you use a whole organism, then you've got something that's biologically true. And that's a good thing because you know that what you're getting isn't a red herring. But again, if you take a whole Babesia microti organism and a whole Babesia duncani organism and you run IFAs, we don't have enough understanding of what the immune system is recognizing on those two different books to know how much of what we're seeing is specific to microti versus duncani and how much is cross-reactive. And so it's very complicated because we're limited by what we know about the biology of Babesia. So like I said, NIH needs to step up and fund more Babesia research and help everybody out with trying to solve the problem of how we detect these pathogens.
Fantastic. With that, if there's no other questions, I want to thank all of you for taking time out of your day to joining us here and having this conversation. So, if there's any questions that you just didn't want to ask live, but you want to shoot us over an email, by all means, please do so. The email is contact@galaxydx.com, and we'll go ahead and get that answered for you. But thank you all. Jen, thank you.
Thank you for your time, everyone.